welcome to another episode of Max Planck Florida's Neurotransmission podcast. I'm Lesley Colgan. I'm joined today by co-host and my dear friend, Anant Jain. Anant, I'm really excited to be here today.
Me
It's
too.
my first episode hosting the podcast, and we get to talk about the brains of octopuses. Did you see My Octopus Teacher?
No, what's that?
Oh, it was great. It was this Netflix documentary about this kind of lonely man who makes friends with an octopus. Every day he dives down and sees the octopus.
Ooh,
You should definitely check it out. I hear they have 9 brains. Well, actually, as a neuroscientist, I shouldn't say that. 1 central nervous system and maybe distributed clusters of nerve cells in each of their 8 arms. It's pretty cool. It allows them to sense and control their arms autonomously. So I'm really excited about the octopuses, of course. But our guest today studies not only octopuses, but all sorts of interesting water animals to uncover how organisms adapt to their unique ecological niche. Our guest today is Dr. Nicholas Bellono, who is an assistant professor of the Department of Molecular and Cellular Biology at Harvard University. Dr. Bellono, we're really excited to have you as our guest here today. Thank you so much for joining us on the podcast.
Yeah, great to have you.
All right, thanks.
So, as I mentioned earlier, you're really broadly interested in understanding the mechanisms that give animals their unique behaviors, such as electroreception used by sharks, or maybe the stinging behavior of sea anemones. So that must mean you've worked with a lot of different animals.
Yeah, so we've had something like 100 species come through our lab. It doesn't mean we work on 100 species at this moment. In fact, most of them we don't end up working on. So our model, our research approach is just to try around and try studying things that we think are broadly interesting. And so students or postdocs will come to the lab. Maybe they have ideas based on, I don't know, something as simple as an animal they've seen in a nature documentary or something that they think is just a very interesting trait. And we stumble upon other animals through, comparative biology, observing something in one species. How does another do it? So there are many ways in which we land on one of these 100 species. And then the next step is to see if we can obtain them, if we can maintain them, and then if we can even technically study them. So for one example, our most recent resident animal model is piranha. And so we've just managed to get piranhas. We had to go through this whole series of permits and, a bunch of trouble to get piranhas in the lab. And they're actually wild-caught piranhas from Amazon.
Wow.
And so we have them now in Massachusetts, one of the few places who can have piranhas, it turns out. And then our next step is, okay, we were able to get these piranhas. How do we, normalize them, make them behave as they would in the Amazon? And can we produce what we think is the interesting aspect of the piranhas, which is, of course, the feeding frenzy that we see in movies where the piranha gets hungry and just tears apart some fish. And so then we see, can we put the piranha in a state where it's fed and it's no longer hungry, and maybe it wouldn't do this feeding frenzy behavior, or can we starve them and reproduce what we think would be interesting in the wild? And so that's kind of where we are with this new model organism in our lab, is to see, can we produce the most interesting phenotype? And now that we have, can we go study and figure out what is the mechanistic basis for what triggers that interesting trait, and why is it specific to the piranha? And that's the same thing like what I talked about here earlier today with octopus. How does it taste with its arms? Can we observe that in the lab? And then can we go and find the molecules and cells that give it that ability?
It's very interesting that you are talking about bringing the piranhas from the Amazon. And do you create some kind of naturalistic environment for them to observe the behaviors? How would they behave in the wild, not just for piranhas, but for other animals, octopuses as well? Or
so we try to varying extent. So for the piranha, well, first I'll say that I don't know how to do any of this. And so I'm really fortunate to have 2 really great people who work in our lab, Brittany Walsh, Peter Killian, who figure out how to do all of this. Basically, they manage the animals, they figure out the aquatic systems, as you can probably tell, they're constantly changing based on what we have. And so they figure out how to house these animals. And our approach is to take the simplest route we can. Can we house them in sort of a minimalistic way? So, for instance, our octopuses live in coffee mugs. They're actually caught in pots. The diver who catches them for us lays down clay pots attached to a rope. They crawl into the pot, he pulls them up, and then he ships them to us. And so we recreate that in their tank, which we, of course, reinforce with some Velcro covers and bricks on the top so they don't escape. But otherwise, it's pretty minimalist. They're in that coffee cup. They have relatively little in their environment. The piranha, on the other hand, requires a little bit more. And so Peter, who set up the piranhas, had to figure out, what makes a piranha a happy piranha to do this behavior that he wants to study. And so he tried to recreate some aspects of their native environment where the water is a little murkier. There's some cover. There's like some wooden logs in there for them to hide under because they're generally actually pretty skittish fish until they're not. So it's pretty variable.
So you mentioned that, you had to try to figure out how to recreate this environment in the lab. It must be really challenging to be continually bringing in these new model organisms into the lab. Do you have to totally come up with all the protocols to do sort of basic things that we take for granted in the lab, like dissociate cells, which might be different in each new model organism?
Yeah, this is definitely the challenge of our work. This is the majority of our time, is figuring out how to do something as simple as you just said, dissociate cells. How do we dissociate cells can take us the majority of the project, right? We have this great example of this where we're studying these photosynthetic slugs. The slugs are awesome. The slugs eat algae and then they retain the chloroplast from algae, which continue to do photosynthesis even when they're in the slug. So it's a great system for thinking about the evolution of really acquiring new organelles. How does one build new cell types, so on. And so our idea was to take the chloroplast from the slug and see what kind of proteins are in the chloroplast once they're in the slug. How does the slug maintain these chloroplasts? How do they keep doing photosynthesis? And so it seemed like a pretty simple experiment, at least if one goes into the literature and sees how do we isolate chloroplasts from plants, for instance. It's just a very simple gradient. You can pellet the chloroplasts out from any tissue you want. And so we did that in the slug. And, long story short, we spent like 6 months studying slug mucus because the slug makes so much mucus that we couldn't even pellet chloroplasts. And so, in order to ask our question, we have to go through this whole process of prep development, which I personally think is great, but some don't. So that's really what happens. And it's true too, for what I talked about earlier with dissociating octopus cells. That was kind of the major step that had to be taken in order to ask these questions about how the cells work.
And in spite of these challenges, it's pretty impressive that you have these variety of techniques that you use, from electrophysiology, imaging, to all the single cell RNA sequencing and those kind of things. It's amazing that you were able to do that with octopus.
Yeah, I think what's nice about developing these systems is you don't know what direction they're going to take. So we have to be open-minded to what we observe, but also open to using new approaches and using new methods based on what we observe, because the question can change because we don't really have a preconceived notion for how this should go or how what the study is going to produce.
So how did you first get interested in working with aquatic animals?
Yeah, a lot of people ask me now, why do we work on marine animals? And the answer is that I really don't even care that much about marine animals. We just kind of ended up doing this. And our lab is actually set up to house other animals as well. We've had plants through the lab that we studied a little bit. I would really like us to study amphibians. We'll see if it happens. But yeah, we're kind of open to any unique trait, basically.
You started your career even as when you were undergraduate. You started a career as working in marine biology lab, and from there on, it's I think your love for aquatic animals just stayed on even till postdoc, right?
Yeah. So when I was an undergraduate student, I spent summer at the MBL in Woods Hole, and we worked on aquatic salamanders in the lab I was in. But then I was exposed to this community where people really do go to the MBL to do really curiosity-based research, study weird organisms, find new things, and I just thought it's so great that someone can do that for their job, and so I wanted to keep doing it. And I went to graduate school and learned a lot about ion channels, biophysics, cell physiology, and then in my postdoc wanted to kind of merge those interests and explore, interesting systems such as electric field sensing in skates and sharks.
So as a postdoc, you worked with David Julius at UCSF, correct?
Yep.
Some of our listeners probably know he won the Nobel Prize in Physiology of Medicine last year for his work on identifying trip channels, which can sense temperature. I found it really interesting that you actually worked on two very different projects in your postdoc. This one that you sort of mentioned about electroreception in sharks and rays, but also did some work on enterochromaffin cells, the cells that sort of line the gut and help transmit signals from the gut to the brain. It's maybe a little bit unusual to work on such different projects. I wonder how you ended up choosing to sort of shift in your future direction away from the gut-brain axis, or if they're more related than I think they are.
Yeah, maybe going back to being a postdoc in David's lab. First, David is a great advisor, and I think gives students and fellows in the lab a lot of freedom to explore. And because of that, people do a lot of different things. And of course, the unifying theme is sensation. But for me, I guess what I really like about sensation is sensory biology is cool, and thinking about how we experience the world is great. But I also just really like cell biology. And for me, sensation is so great because there's a defined stimulus, and somehow that stimulus, whether it be in the environment, in the gut lumen, wherever, that stimulus is transduced to a biochemical event in the cell. And this is why I really like ion channels. It's such a nice model for looking at how that happens. We can make such precise measurements. We can understand, how does one stimulus do this? What about if the cell is presented with multiple stimuli? How are things integrated? And so for me, I really like this idea of, as I mentioned before, exploring animal models and trying to understand that concept in context of an interesting behavior. But of course, that behavior is, or the idea of integrating signals is found in a variety of systems in neuroscience and beyond. And so the gut was really nice in that sense. There was another postdoc in the lab at the time who was studying pain and hypersensitivity in gut and looking at the sensory neurons that innervate gut. But it was very apparent in hearing about how these studies went that there's so much that converges in the gut, ranging from what's consumed, what's in the lumen, the microbiome in the lumen, the sensory neurons themselves, the enteric nervous system, the immune system, like the list goes on, right? There's so many things happening at the gut epithelium. So how does cells in that epithelium transduce those stimuli and do it in a way that makes sense for the nervous system was really what I thought was cool. And that's, to me, not so different than the electric field sensing of sharks and skates, really. So I guess that's how it happened. It's just that broad interest.
Yeah, it actually, that work was very interesting with the electroreception work with sharks and the skates, because you showed how just one mechanism solves two different purposes in these two different species. You want to elaborate a bit on that? Like, one was to detect the prey, if I'm not wrong, and one is to just detect the conspecifics or their own.
Yeah. So this is a hypothesis that we pose. Okay. I don't know if this is true, of course, but what we do, or what we saw was that in skates, there's electrical tuning as such that a stimulus can produce electrical activity in the receptor cell that's rather subtle but shifts. And that shift corresponds to those individual receptor cells releasing vesicles or synaptic vesicles at different rates. And what this means is that then the nervous system will interpret what the receptor cell is doing, right? Depending on what frequency that receptor cell sees. As where the shark cell, in contrast, produces large action potentials to any stimulus that we gave it in the range that those channels are active. So this was more like an all or none trigger. And so our idea was that maybe, and it took us a while to kind of come to this idea, maybe the reason for this is that the skate needs to interpret more precisely electrical frequencies because the skate does have an electric organ where it produces electric discharge, and other skates will detect that discharge to find mates. But it also uses it for predation. So maybe there needs to be this kind of tuning within skate, as where shark does not have an electric organ, and it's really just using it for predation. And so that's the model we posed. And the one thing that we did to test that, I worked with another postdoc in the lab, Duncan, who did this behavioral experiment, which I thought was great. We thought of for a while, what could we do to see, like, I don't know, sharks attacking things or skates doing something interesting. And we came up with the, I think, best experiment that Duncan did, which was to look at the ventilation rate. And I always liked giving this presentation because I'll do the whole story about the ion channels. Then, of course, people want to see a shark eat something, right? So I get to the very end, and then what we show is the gill movements of the shark versus the gill movements of skate. And we can give them these electric fields, or we can give them odorant. And what the fish do is, once they're exposed to the sensory stimulus, they'll change the rate of ventilation. And this is sort of a sign that they're interpreting this stimulus, or they're getting excited about some fish in the water or something. And so the skate, you can see a nice tuning curve where if we give it different frequencies, we'll get more or less ventilation responses. With the shark, it's like a very similar, just almost identical ventilation response to any stimulus. So I thought that was really a funny experiment for, like,
Super
we're talking about predation in sharks and we watch their gills move.
Yeah, that's great. I wish I could see that video too. It reminds me of the awesome videos you showed today during the talk about octopuses. And I'd love to talk more about octopuses and what you found there. So maybe you can just give our listeners a recap of sort of these new sensory receptors that you identified in octopus and their sort of unique role in what you call the taste by touch.
Yeah, so as I described for taste by touch, we found a mechanoreceptor that's responsible for the touch component. And then for taste, we characterized this family of ionotropic receptors that was first identified in the genome because they look like acetylcholine receptors or nicotinic acetylcholine receptors, but they don't have the canonical acetylcholine binding site. And so we found that those receptors were greatly enriched in the sensory epithelium of individual suction cups on the arms. And so we went through and characterized a couple of those to see, are they involved in chemoreception? And how we did that was comparing the native chemoreceptor cell or system with cloned receptors in expression system to see does the biophysical properties and pharmacology match? Is this really representative of what the octopus is detecting? And what was very surprising, and we found sort of in a weird screen, so we've actually found what the receptors detect by screening basically whatever we could find around our building. So we took the receptors, we put them in expression system, and we did that to ask, what does the octopus detect? Because we actually didn't know that up to that point. We were using ground up prey as a stimulus just to broadly test, what could the receptors possibly be? What are the cells? But we didn't know the individual molecules. So the receptor screen serves sort of two purposes. One, to compare with the native system, is the pharmacology the same? But then also to define, what does the receptor detect? What does the octopus actually probe with its arms in terms of individual molecules? And And so we did this receptor screen by taking what are sort of standard tastants, what one would use to probe chemosensory systems more broadly, like amino acids and so on. And none of those did anything. But what was very surprising was what was active on the receptor are these molecules called terpenes. And terpenes are broadly found in plants and microbes largely. And we thought that that made sense and has actually been posed as an idea for this contact-dependent chemosensation that octopus and other aquatic organisms do, because terpenes aren't as soluble. So the idea is that they would be sort of near the surface that the animal needs to probe. And so that sounds great, but actually how we found terpenes was a colleague down the hall, Venki Murthy, has just a ton of odorants, tastants, or things one would use in a chemosensory screen. And we went and raided his freezer and then just screened every molecule we could find. And a bunch of these terpenes were active. And so now we're finding that that sort of random experiment ends up being, at least as far as we can tell, sort of in the right direction, in the sense that we find that those molecules are active from the individual receptor proteins to the cells, to the nerves of the arm, to producing behavior. And now, as I presented today, we see in the structure of one of the octopus receptors that these greasy molecules, even greasy molecules that we find by accident, are actually found in this adapted, very hydrophobic pocket that's so different than the nicotinic acetylcholine receptor.
Evolutionary speaking, how different is this mechanism from our taste buds? And is it changed because of the environment we live in? What do you think? Have you given some thought on that?
Yeah, so they're pretty different. They're ionotropic receptors, as I described today. And what that means is that they form ion channels themselves, as where our taste receptors are G protein coupled receptors that trigger this cellular signaling cascade that eventually results in ion channel activity and then cellular excitability and some signal that's transduced to the nervous system. For the octopus, it uses the ion channel directly to detect the chemical and then to transduce the signal, because it gates the ion channel, allows ions to pass into the cell, that produces the electrical signal. And so at least how we're thinking about this, and of course this is our story of how evolution happened, but we have no idea, is that we think the ion channel makes sense because it's actually a pentameric complex. So 5 proteins come together to form the channel pore, the ligand binding site, so how it detects the chemicals. And these 5 subunits can be different. So you can have a homomeric channel, which means 5 of the same come together, or very different subunits can come together. And that changes which ions can go through the pore, how many go through the pore, and then what kind of molecules are detected. So our idea is that maybe this is well suited to the octopus system because it gives this diversity, not only for signal detection, because it is using this sense for exploration, but maybe also for transduction in the context of this distributed nervous system that does a lot of processing in the periphery. Maybe processing even starts to happen based on how they can assemble those complexes.
Super interesting.
Yeah. So in thinking about sort of these differences between our sense of taste and the octopus's sensory organ, we know that our sense of taste is really highly influenced by things like smell and other sort of higher order processes. Do you think they're sort of similar higher order processing of the sensory information that's coming from these receptors in the octopus?
Yeah, I think this is probably the case. I don't know how it works or what influences it yet. But what I can say is that we do these experiments where we try and measure from nerves to see if these signals are transduced, the way we were just discussing. And what we see is that even at the level of the arm, if we apply these molecules which stimulate the chemosensory system, the arm will produce autonomous behavior. The arm can itself independent of the octopus's centralized brain, it can produce reaching motion, it can retract, it moves quite a lot in response to stimuli. So there's sort of that level of autonomous processing that probably takes account of both touch, this analogous form of, I guess, what we're calling taste, where I assume a number of molecules can converge and produce different behavior. But then, of course, the animal is a very visual predator as well. And while it uses the arms, which again can do this relatively autonomous behavior, it uses arms to probe the cracks and crevices of the seafloor for prey. It can use its visual system quite well to find prey. And so I'm sure that these two things converge and probably in very interesting ways to control the arms so that they're in unison go, attack and pounce on prey if it's visual or not. So I'm sure all this stuff comes together in some way that I can't begin to imagine.
Talking about higher order processing, I was wondering if you've observed some kind of learning or habituation where these chemoreceptors are involved and in these arms, behaviorally and also mechanistically.
Yeah, not yet. The only I would say some experiments that we've done in that direction are to ask if the receptor complexes change in response to some kind of context of the animal, and maybe that's I guess higher order in the way that the cell is regulating the expression of these subunits or some kind of modification of the subunits. So we've been thinking about that, given that the receptors can combine in these very distinct ways and produce different biophysical properties. Does the octopus tune maybe one subunit based on being hungry? Maybe it needs to detect, I don't know, a molecule that tells it a crab's nearby more sensitively than if it's not. Or that's our idea, but I don't have an answer.
One cool thing about octopuses is that they can regenerate their arms, right? And
Yeah.
I was wondering, like you talked about these expression of chemoreceptors, some patterns that you found, and do these distributions change when the arms regenerate? Is it environmentally driven? Is it genetic?
Yeah, I don't know. Because I actually don't know anything about the expression patterns yet. It seems like pretty difficult to interpret. They're kind of co-expressed everywhere. Is there some molecular logic to it? It seems like there might be or should be, I guess, but I don't know. And so I think we need to first define fundamentals in order to ask the next question, which I agree is very interesting. If it can regenerate limbs, how does it recapitulate some pattern if there is one? But first, we have to define the patterns. We have to define the receptors. We have to define what they do if they're expressed in a pattern. There's kind of a bunch of fundamentals to describe.
But do you study isolated limbs or arms of these octopuses? Just and how long does it take to regenerate usually?
Um, I guess, yeah, I don't know. I don't remember how long it takes to regenerate an arm. It depends on how much of the arm is lost, I suppose. But they do regenerate, and they can persist pretty long if the arm is isolated. I've heard stories of people putting them in media for like weeks at a time or something. We never do that. We've only studied tips of arms for these nerve recordings that I mentioned, and they're cute. So I don't know the answer.
So, one thing I really appreciate about how your whole research program is set up is that it's very curiosity driven. And here at Max Planck, that's something that, we really strongly believe in curiosity driven research and the idea that sort of insight must precede application. Um, However, in the US, I feel like this model of science tends to be more of the exception rather than the rule. And some people even suggests that our current funding system doesn't always support curiosity driven research, but you've also been very well funded. So I'm just wondering if you could maybe comment on this or why you think curiosity driven research is a good approach.
So I could give a few answers to this, something theoretical and something practical. So in theory, I think curiosity driven research is a good approach because one never knows where the next breakthrough comes. One never knows how, as you described, something very basic can turn into something very applied. There are countless examples from this: GFP, CRISPR, blah, blah, blah. So we could talk about that. In practice, I think curiosity-driven research is important because, that's why I'm a biologist. That's why I like doing this job, because I get to study biology, and I would like to study biology that I think is cool. And it's such a privilege that we get to do that. So I think that we should emphasize being able to do that and find ways that we can instill uh, love for science in trainees, and um, uh, I don't know, a fearlessness and bravery to go into new territory and study new things. That's how we learn new things, and that's one of the reasons why I think it's important that we do that, and why I think it's important that my lab does that, is that people can learn that approach, because I think that's the most fun part about being a scientist. So that's in practice why we do that. And then in terms of funding, what do I think about this in context of funding? We've mostly been successful with it so far, and I think the reason for that is kind of related to what I just described, which is that I believe that this is a good approach for learning new biology, because we're looking at new questions, new systems, and that's how one learns something new. And so, what I've always talked about in the context of funding, or in our applications, is um, basic principles. We learn a lot about basic principles. You mentioned this sea anemone stinging, which I think is such a good example of this, where we made this really unanticipated discovery of how this individual stinging cell, which is this cell that houses this very odd organelle that has this really long thread that's attached to this stinging barb that's coated in venom, and the organelle will shoot out at the rate of a bullet this thread with the barb to sting prey or predators. And it does it only in response to chemicals and touch, not touch alone. And this is at least how it's presumed that jellyfish and anemone can distinguish between prey or mechanical disturbance of a wave or their own tentacles. And so we studied that system because it's really such a nice system for thinking about how does one cell integrate these signals and make the right decision at the right time. And we found this very unusual ion channel that can integrate these signals. And to me, it was just a really nice example of a basic principle of what I just described. How does a cell take in signals and produce behavior? So I think, or I hope, that our funding institutions value basic science and basic principles. And so far, I've been pleasantly surprised that that seems to be the case. Review panels do respect that aspect, that we don't know where new discoveries can come from, and the only way we're going to make them is if we look.
And it's great that you're able to set a good example for this curiosity driven research, because this is very important. This is actually the core of doing research, is to like have this freedom of just observing something interesting in nature and then trying to understand how this is happening. This is very impressive. So, and I'm glad that the funding agencies are recognizing that now. It's very, very cool.
Yeah, we have fun. It's mostly fun. It sounds fun. It's also hard, as I described.
Yeah.
There's no sort of protocols in place, or it's a learning curve for trainees in the lab for sure. But I think it's a great way to learn, because they learn a lot about protocol development. How does one pick the right system? How does one know when to keep going and not?
And not get limited by the species, get limited by the techniques, right? So that is the great part of doing work in your lab. Yeah.
Okay, so we're almost out of time, but before we let you go, I just wanted to ask if there is anything new in the lab that you're really excited about right now.
So I'm excited about most things in the lab these days, honestly, which I think is a great situation to be in. But one thing I showed in this talk today that we haven't discussed, that I think is worth mentioning in the context of curiosity-driven research, is this really great fish called sea robin. So we first found sea robins when we went to the MBL in Woods Hole to pick up cuttlefish, I think it was. And we saw these fish because one of the guys who works in Marine Resource Center, who I've worked with since the days of studying sharks, took me to show this fish. He said, Nick, I know you're into this weird stuff, so you got to see this great fish. And what the sea robin looks like is it's this kind of crazy looking fish that has basically legs. There's these leg-like appendages. They're actually fin rays that split off from its main pectoral fin. And it uses the fin rays to walk around on the seafloor, basically, or crawl around, I guess. And what he was telling me is that these fish find very well buried prey. And so we took the fish back to the lab, and it turns out there's a few studies about this, how the legs are chemosensitive. And so we tested this, and we buried prey at different depths. We buried capsules with ground up prey. We buried chemicals in capsules, and the fish are great at finding those capsules, to the extent that there are actually studies that show other fish will follow around the sea robin, hoping for leftovers from the sea robin's captured prey. So they're really good at doing this. And so Corey Allard, postdoc in the lab, who's studying this, spent a lot of time trying to find the receptors on cells for this behavior, because we thought it's such a great system to think about the convergence of development and physiology for making new organs, new sense organs. So he spent a lot of time looking for these cells, these receptors. He was really struggling. We went back to the MBL, and by accident got back another sea robin. And this sea robin looked pretty similar in the sense that it had these big pectoral fins. It also had legs that it crawls around on, but it doesn't find the buried prey. And so Corey had this really great now comparative model, which he's been able to track down certain aspects that specify this new aspect of this organ, which this fish develops. And it's actually quite specialized to a certain group of these sea robins that have this chemosensory ability and specialized its organ. So I think this is just a really great study in the context of comparative biology, and a really great study in the context of being open-minded and just following observation.
Cool. Looking forward to your new talk on sea robins next.
Thank you so much for joining us on the show.
Yeah, thank you so much.
Thanks for watching today.
If you like this video podcast, make sure to subscribe to our YouTube channel for more cool neuroscience.